Hello, and welcome to the second of the 2024 Gairdner Global Perspective Panels presented by TELUS Health. I'm Sarah Devonshire, the Director of Awards and Outreach at the Gairdner Foundation, and it's my pleasure to welcome all of you here with us today. Over the coming months, we will be hosting a series of Global Perspective Panels led by our Early Career Investigator Competition winners from recent years. Together with presentations from world-leading researchers, they're sharing the latest developments in precision cancer care and omics, prospects for mRNA therapeutics, the microbiome from the microscopic to global, and AI in healthcare. We hope you'll enjoy today's discussion and visit Gairdner.org to sign up for upcoming events. I will now hand over proceedings to Anna Blakeney and Bowen Lee, 2022 ECI winners from UBC and the University of Toronto, to begin the panel. Thank you.
Welcome everyone to our Gairdner Global Perspective Panel on the future of mRNA therapeutics. I'm Bowen Lee, assistant professor at the University of Toronto. I will moderate today's session with my colleague Anna Blakeney from the University of British Columbia. So we are honored to have such a great panel of experts join us today to discuss this revolutionary field. mRNA therapeutics have emerged as a groundbreaking approach with the potential to transform medicine. From the rapid development of COVID-19 vaccines to the development of the personalized cancer treatments, mRNA's versatility and adaptability have opened new avenues for addressing a wide range of diseases. So in this panel, we will talk about the current landscape of the mRNA therapeutics and discuss its applications, challenges, and future directions. Now, Anna will introduce our first panelist today.
Thanks, Bowen. I'm really pleased to introduce our first speaker today, Dr. Yizhou Dong, who is a professor at the Icahn School of Medicine at Mount Sinai. His research focuses on the design and development of biotechnology platforms for the treatment of genetic disorders, infectious diseases, and cancers. Dr. Dong has authored over 100 papers and patents, and several of his inventions have been licensed and are currently under development as drug candidates for clinical trials. He serves as a scientific advisory board member for Arbor Biotechnologies and Cernogen Therapeutics, and Dr. Dong is a recipient of multiple awards, including the Maximizing Investigators Research Award from the National Institute of General Medicine Sciences and the American Association of Pharmaceutical Sciences Emerging Leader Award. Welcome, Dr. Dong.
Hello, everyone. Thanks so much for the very kind introduction. Thanks, Bowen and Anna, for organizing this very exciting global panel, perspective panels about the mRNA therapeutics. So it's my great pleasure to briefly share with you several examples of our work. I really look forward to the afterward panel discussion with another two experts and friends, Christian and Sarah. mRNA has now become a very important class of vaccines and therapeutic agents. During the COVID-19 pandemic, mRNA vaccine was administered to the first volunteer in a phase 1 clinical trial when only like 10 weeks after the viral sequence was determined. So this incredible speed is based on over 6 decades of hard work in discovering mRNA and its properties, as well as critical advances in delivery systems such as lipid nanoparticles, polymers, and other platforms. I was very excited last year. The Nobel Prize in Physiology or Medicine was awarded to Katie Carrico and Drew Weissman. Their pioneering work enabled the development of effective mRNA vaccines. Even my son, he's only 10-year-old, he talks about the mRNA and lipid nanoparticles. Once he asked me, "Are you going to lose job?" I asked why. He said, "mRNA vaccine is already used in people. Anything else you can still work on?" I got similar questions from reporters and also colleagues as well. In fact, I feel this is a wonderful time for researchers in the field of mRNA therapeutics. There are still challenges and opportunities in this field. How to deliver mRNA to specific organs, such as lung, pancreas, bone marrow, brain, and so on. How to create mRNA sequences and chemical modifications to best fit individual therapeutic indication. There are so many organs and cell types. There are so many unmet medical needs. So in the following slides, I will share with you a couple examples. Based on the significant advances in the delivery field from many pioneers, such as Peter Collis, Bob Langer, Nicholas Peppers, Frank Zocca, and many others. And we designed this class of lipid-like compounds, and it has a long chemical name shown here, and we just give it a short name, TT lipids. So these TT lipids are composed of phenyl ring, 3 amide linkers, and multiple lipid chains. And we can synthesize these TT lipids in large scale with this very simple reductive amination reaction. This slide shows our overall strategy for the development and optimization of the lipid or lipid-like nanoparticles for mRNA delivery. And we have newly synthesized TT lipids, phospholipid, cholesterol packed lipid, and the therapeutic cargo here is messenger RNA. And through this microfluidic-based device, we can assemble them into very nice nanoparticles. And then we can characterize the particle properties, such as the particle size, charge, mRNA loading efficiency, or mRNA translation efficiency. And then we utilize different approaches, such as orthogonal experiment design, or barcoding system, or other approaches. We can optimize the formulations of the lipid components, and then we can identify the optimal formulation for disease models. And here we chose a model disease, hemophilia, which is caused the missing or mutation of factor 8 or factor 9. Patients suffer from life-threatening bleeding or serious complications such as joint bleeds. So in this study, we formulated TT3 with mRNA encoding human factor 9, and then we injected intravenously first in wild-type mice. So on the left figure, the y-axis, that's a human factor 9 concentration in mouse plasma, and the x-axis different treatment groups. And you can see it showed a dose-dependent recovery of the factor 9 concentration. Then we performed similar studies in factor 9 knockout mice, which is a hemophilia B mouse model. And you can see the trend is consistent. Next, we are wondering if these factor 9 proteins are active or not. So we performed activity assay. And this y-axis, that's a human factor 9 activity, x-axis different treatment groups. And you can see it's also showed a dose-dependent manner. And at 1.1 milligram per kilogram, the factor 9 activity is around 800 MIU per ml. In normal human plasma, the factor 9 activity is between 500 to 1500. So that means TT3 is able to fully recover the factor 9 level to the normal range. Later on, we collaborate with the groups of Ron Weiss and Darrell Irvine at MIT. We formulated TT3 with self-replicating RNA encoding IL-12 for cancer immunotherapy. And this illustration of the study, we formulated the lipid nanoparticle with replicon, and then we performed the intratumoral injection, which would trigger the subsequent anti-tumor immunity. They performed a lot of in vivo studies, and here are some representative data. With this very simple single injection of the lipid nanoparticle formulation, we can see a very high percentage of the complete response in 3 different mouse tumor models, B16F10, CD26, UMR 1.7. And they also performed quite a lot of mechanism action studies. If we knock down the MYD88 or STING pathways, here the therapeutic efficacy is significantly compromised. Meanwhile, we performed the chemical modifications of the TT lipids. We can incorporate carbon chains, branched ester chains, or linear ester chains, And then we formulate the particles, as I just mentioned. And on the left, you can see this luciferase acid. FTT5 is a lead material, which showed around a twofold better delivery efficiency than TT3. And on the right, these are the particle properties. The particle size are around 100 nanometers, and these particles are relatively homogeneous. We then collaborate with Beam Therapeutics, which is a leading company in the field of base editing. So we formulated FTT5 with mRNA encoding base editor and the guide RNA targeting PCSK9. And on the left here, the y-axis, that's a base editing activity, x-axis different doses. And it also showed a dose-dependent base editing activity. On the right here is one set of data for targeted deep sequencing at a dose of 0.125 milligram per kilogram. And then you can see the editing efficiency is still over 50%. So our industry collaborator is very excited with this activity, and to our knowledge, this is the most efficient base editing ever reported in the literature. So in conclusion, I briefly talk about a couple of lipid nanoparticles, and we can deliver to hepatocytes, cancer cells, and there's some proof of concept studies for the treatment of genetic disorders, or for cancer immunotherapy. And currently, these materials have been licensed by several biotech companies, and some are in clinical trials, and some are in IND-enabling studies. So for the next few years, we think we need to integrate specialties together with our collaborators from different fields, such as biomaterials, RNA engineering, genome editing, immunoengineering, and cell therapy. And we really hope to translate our fundamental findings and the technology developments into more effective treatment for the patients in the clinic. Here's my disclosure. And lastly, I would like to acknowledge a lot of people. We have a group of talented postdocs, graduate students, undergraduate students, and a few high school students in our group. And thanks our collaborators for their significant contributions for many projects. And also thanks the previous and current funding agency for their generous support for our very different, very diverse projects. And thank you very much for your attention. I really look forward to the following panel discussion. Thank you.
Thanks so much. It's really fascinating to see all the new applications that you're applying mRNA therapeutics to. So next, I'd like to introduce our next speaker, Dr. Christian Kastrup, who is a senior investigator at the Versiti Blood Research Institute and a professor at the Medical College of Wisconsin. His appointments are in the Department of Surgery, Division of Trauma and Acute Care Surgery, and the Departments of Biochemistry, Biomedical Engineering, and Pharmacology and Toxicology. Additionally, he's an affiliate professor at the University of British Columbia and is a co-founder of Nanovation Therapeutics, Serogene Therapeutics, and CoMotion Drug Delivery Systems. Christian is a biochemist specializing in biotechnology and genetic medicines. He earned a PhD in chemistry at the University of Chicago and completed a postdoctoral fellowship in bioengineering at MIT in the lab of Dr. Bob Langer. His research is centered on understanding and controlling blood coagulation and fibrinolysis proteins. He is driven by the potential to improve health outcomes of young people by addressing the impact of hemorrhage during trauma and childbirth, as well as thrombosis-related conditions such as strokes and deep vein thrombosis. In his laboratory, Christian's team creates RNA therapeutics, lipid nanoparticles, advanced drug delivery systems, and new blood products for transfusion medicine. Christian has trained and supervised over 100 scientists and clinicians in his laboratory with these shared goals. Welcome, Dr. Castro.
Thank you very much, all of you, for tuning in, and Dr. Blakeney, thank you very much for the introduction and for allowing me to present today. So as mentioned, I'll be telling you about how we can use mRNA therapeutics for, in this case, a particular set of applications related to blood disorders or problems with blood. And as Dr. Blakely mentioned, I have a few disclosures I'm required to make. Affiliations with Nanovation Therapeutics, Saragene, Comotion, and consulting or grant funding with these others. So a problem that I'm very passionate about solving, that my laboratory is very passionate about, is bleeding. And we can all relate to bleeding in some way, probably many ways. Here's one. Here's a severe hemorrhage after involved with trauma. But bleeding, of course, is an issue in many ways. So we're interested in this because trauma is the leading cause of death of young people worldwide. And by young people, I mean below the age of 45. Of course, it's a problem for people that are older than that as well. Just to give you a sense of the issue, about 2 million platelet units are transfused annually in the US. So platelets are transfused to prevent bleeds or to stop bleeds. And so there's 2 million of them. So there's a lot of bleeding that's happening. In addition to trauma of otherwise healthy people, bleeding disorders are a common problem. Dr. Dong just described a bleeding disorder, so hemophilia B. And so today I'll be telling you about nanomedicines that we've been developing to control proteins in the blood and bone marrow and to improve blood. And so, like Dr. Dong, we really utilize lipid nanoparticles in order to deliver genetic medicines to the liver. And so lipid nanoparticles, if you inject them into the muscle, into your arm, it'll have a vaccine-related response. But if you inject them IV, they can go to tissues that take up these lipid nanoparticles and deliver their contents, in this case, RNA. And for the most case, they mostly go to the liver. We'll talk a little bit more about this at the end. But the liver is a great place if you're interested in controlling blood, because most blood proteins are made in the liver. And so you can deliver lipid nanoparticles containing small interfering RNA, which will decrease the concentration of specific proteins in the blood. You can deliver mRNA to produce proteins in the liver using it as a bioreactor. And you can deliver mRNA for CRISPR reagents like Cas9, along with a guide in order to permanently cut genes in the liver or to modify specific bases. And so we use lipid formulations. They're developed by Peter Collis at the University of British Columbia, as well as Marco Cifalini. These are similar formulations to those that were approved by the FDA in 2018 by Alnylam for an siRNA therapy, and in the 2 SARS-CoV-2 vaccines and the recent RSV vaccine. And so we're lucky that these lipid nanoparticles naturally go to the liver, because most proteins in the blood are made there, including the proteins involved in coagulation, and which is the formation of blood clots, and the degradation of blood clots, or fibrinolysis. And here's just a small portion of the proteins and enzymes involved in blood clotting. There's a lot of them. This may be a third of them. Most of these are flowing in our blood, I should say. And the ones that are circled in green or have a dashed green circle around them are ones that are made in the liver. And here's an example of one I'll talk a little bit more about. This is coagulation factor 7. And so in an application very similar to what Dr. Dong was mentioning, in people with bleeding disorders such as hemophilia, they're missing a protein in their blood. And so here we can replace that by producing the protein with mRNA. So you take lipid nanoparticles, the mRNA in them, inject them IV, they go to the liver, and the liver produces the missing protein. And so if you look at the amount of mRNA in wild type mice, has a normal amount of this mRNA, in this case for factor 7. If the animal has or human has a hemophilia, they'll have less of this. But by delivering the mRNA therapeutically, you can increase that and bring it back to normal. And that can normalize the amount of blood loss when there's an injury. So a wild type mouse won't bleed very much. If it has this disorder, it'll bleed more, and you can correct it by delivering this mRNA. And so this will last a couple of days. But one area that's really important growing area for mRNA therapeutics is being able to extend the half-life. And this is a collaboration with Dr. Blakely. And so there are many approaches being developed in order to deliver mRNAs that will, or mRNA-like molecules that will last much longer. And so this is a case where delivered this RNA for factor 7, we can prolong its expression for over a month. And so, so Dr. Dong and I, we told you about replacing proteins in the blood or enhancing proteins in the blood. Another approach we can do to improve our blood is a cell therapy approach. So take the cells ex vivo, modify them with mRNA, again with lipid nanoparticles, and transfuse these cells back into the bloodstream. And as a blood clotting lab, we focus a lot on platelets. And so what we're really trying to do is create transfusible platelets to locally deliver RNA and proteins from those platelets. And so if a person is transfused with platelets, these platelets naturally circulate and they get deposited. They accumulate in areas of damaged vasculature. And so if we can modify those platelets, we could deliver the RNA as well as the proteins to areas of vascular damage, like areas of hemorrhage, areas of thrombosis, where there's blood clots, to cancer, areas of inflammation, and angiogenesis and wound healing. And so we hypothesized that we could take platelets, donated platelets to a blood bank, transfect them with lipid nanoparticles and mRNA, and express proteins in these platelets. They're in some ways they're very amenable to this because they don't have nuclei. So other approaches to genetically modify them with DNA, with viruses, won't work because they don't have nuclei. And so we hypothesized that we could take lipid nanoparticles, and we can do this. So we can take a bag of platelets, inject the mRNA lipid nanoparticles, have the platelets take them up, and then produce proteins that they didn't have already. And so if you transfuse these platelets, and here the platelets are in yellow, you can see them accumulate in areas of vascular damage. So what we're looking at here is a kidney of rats that are transfused with modified rat platelets. Usually we modify human platelets, in this case rats. And there's a cut that's made here. And in the red, you can see all the platelets accumulate, normal platelets. And the platelets that are modified have been edited with the mRNA. They show up as yellow. So they accumulate in this area of damage. And they're essentially delivering this mRNA and protein to that area. And so coming soon, in this field, really is being able to deliver RNA to a wide variety of tissues. So it's easy to get them to the liver. There's plenty of opportunities, but really expanding beyond that to other tissues. And it's important which models are used, which animal models are used for this. But what we find is that with pretty standard mRNA lipid nanoparticles, you can inject these in large animals. You get some transfection, some uptake of the mRNA and protein production in a wide variety of tissues, pretty much most tissues. And here's just an example of being able to transfect the bone marrow and cells in the bone marrow in order to get protein expression there. And so that's a portion of what I wanted to share. And many collaborators that contributed to this, including Dr. Blickney, and trainees, especially Jerry Leung and Francesca Ferrasso, started this work. So yeah, thank you very much again for attending and allowing me to participate.
Great. Thanks so much, Dr. Kastrup. It's really exciting to see all these interesting approaches for treating blood disorders. So next, I'd like to introduce our last speaker on the panel, Dr. Sarah Hedtrich, who obtained her PhD in pharmacology and toxicology from the Freie University Berlin in Germany in 2009. During her postdoc, she moved to the Ludwig Maximilians University in Munich and Tufts University in Boston. She was appointed as an assistant professor at the Freie University in Berlin, Germany in 2015, and then relocated her lab to the University of British Columbia in 2019. Currently, she holds one of the prestigious Johanna Quandt professorships at the Berlin Institute of Health at Charité in Berlin, Germany, and is an affiliate professor at University of British Columbia. She has co-authored over 95 peer-reviewed journal articles in high-impact journals, including ACS Nano, Journal of Controlled Release, Small, Nature Review Materials, and Theranostics. Her research centers around inflammatory and genetic diseases of human epithelia with a focus on skin and lungs and bioengineering of complex human disease models, which are leveraged to develop personalized next generations therapies. Welcome, Dr. Hedrick.
thank you very much for the invitation and for the kind introduction. And I'll go ahead and share my presentation now. So I want to switch gears a little bit in my presentation. You've already heard a lot about LNP delivery systemically to the liver and for mRNA or for protein replacement therapies. And what my lab is interested in is really how we get the mRNA therapeutics into very restrictive tissues. And my lab is really focusing on human epithelia and specifically on human skin and the lungs. And the overarching focus of my lab is that we are striving to tackle and understand inflammatory and genetic diseases of those epithelia. And in order to do that, we are developing complex human-based disease models, which we then use to study disease-related mechanism, but also for the testing and development of next-generation therapies. And that's exactly what I will focus on in today's presentation. And we are using those human disease models because we aim, or we really believe, that this eventually facilitates translation from bench to bedside, which is one of our major goals. So when thinking about human epithelia, like the skin and the lung, there are certain specifics about that. I mean, they are in direct contact with our environment, and they are basically securing our survival. And as such, they have developed very restrictive epithelial barriers, which are quite distinct. And in the human skin, for example, here you see a cross-section of human skin. This outermost very thin layer, which is the stratum corneum, basically prevents us from dying because it prevents massive transepidermal water loss, and it also prevents that pathogens can enter the skin, which is great, in order to secure the survival. However, when thinking about delivering drugs, and especially gene therapies like mRNA therapeutics into the skin, this really causes problems and makes it very challenging. On the other hand, the other organ we are interested in are the lungs. And in the lung, we have a very different biological barrier, which is the mucus layer, which is on top of the cells, and also the cilia, which are continuously beating and moving the mucus forward. And again, this prevents especially biomacromolecules like mRNA from efficiently being taken up or entering the bronchial epithelium. And so some of you may think, okay, when targeting the skin or the lungs, why don't you go systemically? And I know there are a couple of approaches and people testing that. However, I truly believe that the most promising approach is to do it topically. And I'll give you an explanation for that, because, when you inject, for example, lipid nanoparticles that contain mRNA systemically, then it definitely enters up into the lung. And there's great data showing that there's certain lipid nanoparticle formulations that have a preference for lung endothelium, for example. But in order to really reach the lung epithelium, what would need to happen is that those lipid nanoparticles need to migrate out of the blood vessels and then somehow traverse through the extracellular matrix over the basal membrane and then transfecting the epithelial cells. And I really have a hard time imagining how this is possible. This is different in the alveoli, but when thinking about the bronchial epithelium, it's very hard to imagine, and the same holds true for skin, which, for example, does not contain any blood vessels in the viable epidermis. So my lab is really trying to develop strategies to deliver mRNA-based therapeutics, and in our case, we are working on CRISPR-based strategies in order to provide a potentially curative effect topically. And I'll give you a few examples of the work that we are doing. So with respect to skin, we are very much interested in developing the first effective and potentially curative treatment options for patients that suffer from genodermatosis. And as you can easily appreciate, that these are highly stigmatizing diseases. They are very rare diseases, and in some cases, they are also life-threatening. And there's a huge medical need because there are no effective treatment options. Now with the new mRNA-based strategies, including also CRISPR-based gene editing, we do have the tools to effectively treat those diseases. And the approach that we are taking is that we are utilizing lipid nanoparticles, which are excellent carriers for mRNA and gene editors. And then we apply them topically onto the skin, onto the diseased skin. And even though the skin is diseased, it's still, is a sufficient barrier to prevent lipid nanoparticles from efficiently penetrate into it readily. And so what we need to do is to really modify the epidermal layers. And we do this through microporation or microneedling, with the goal to really reach this one single basal layer of stem cells in the skin, because when we reach those, we can effectively cure the patients, because from there the skin regenerates every 4 weeks. And so we really have a chance to provide an effective and curative treatment to those patients. And over the past 5 years, we've made significant progress in that regard. And I don't have the time to go into detail, but I can tell you that we established different dose response curves, and we are, as I mentioned at the beginning, exclusively working with primary human cells and tissues. So we are collecting freshly excised human skin tissue, which we get from plastic surgeons. And then we have the best model possible to assess or to determine the editing efficacy. And so when you look at those editing rates that we achieve in a dose response, dose-dependent manner, you may be disappointed looking at only 10 to 15%. But I can tell you that there's data showing that even if you can only rescue about 5 to 10% of the gene function, this suffices to alleviate the most severe symptoms for the patient. So we are definitely in the correct range, and this is very promising, and we are pushing that forward. When thinking about the lung, it's a little bit different, because we are facing a completely different barrier. And I already mentioned the mucus layer, which is already a very restrictive barrier in healthy conditions. However, there are certain genetic diseases, including cystic fibrosis, where there's no normal mucus layer. You have, in fact, a very highly viscous mucus layer with high DNA concentrations. The cilia movement is impaired. And there it's even more challenging to deliver lipid nanoparticles or mRNA therapeutics through this mucus layer to the epithelial cells. And just to illustrate that is we tested how lipid nanoparticles perform in normal lung mucus and in patient-derived mucus. And I'm playing some videos now, and I think it's quite impressive. What you can see on the left-hand side, those bright little dots which are dashing around, these are the lipid nanoparticles in normal lung mucus. So they are quite happy and move a lot. On the right-hand side, we collected patient samples, and so the video is actually playing, but you can still see those dots, but they are not moving at all. And so this is really a challenge that we have to overcome when aiming to deliver lipid nanoparticles and mRNA therapies to the lungs of cystic fibrosis patients. And so, in the past, those challenging biological barriers have been disregarded or neglected. And so it was no surprise, honestly, to see that early studies from certain companies that aim to deliver mRNA to the lungs of cystic fibrosis patients fail, because when looking into the data, it became quite clear that, they neglected the challenging barrier properties, and also the preclinical models they chose to assess the efficacy and delivery efficiency of their approach were not entirely suitable. So I think we can do better. And so we really, we need to do better in that regard in order to make this happen. Yeah, so these are those two angles that my lab is working on. And especially for the skin part, we are pretty much advanced. We are already in discussion with regulatory authorities, and we are working toward getting that into the clinic by the year 2027. So in my opinion, these are really exciting times. And, the mRNA-based therapies, they hold a huge potential. And hopefully this will unfold over the next decades. Thank you very much.
Great. Thanks, Dr. Hedrick. I really enjoyed hearing about using RNA therapeutics specifically for targeting the human epithelium. So we will now move on to the panel discussion, and I'd like to invite all the attendees to submit their questions if you have any through using the Q&A function. All right, so maybe we'll go ahead and get started with the first question. So we heard about some really diverse applications from all of our panelists. And I thought we're planning on using RNA therapeutics for. And I'm wondering if you can all discuss a bit about how these are an improvement over the current treatment options and how you really see this making a difference in patients' lives.
Yeah, maybe I can start, at least for the diseases that we are working on or interested in, especially the skin diseases. There's, as I mentioned, there's no treatment available at all, or at least no, it's purely symptomatic, and very often it doesn't really improve the situation for the patients. And we already have seen, I mean, not in the context of rare skin diseases, but in other rare diseases, that mRNA-based therapies can really make a huge difference. And so I think there's a lot of hope to change the lives of many patients for the better using this technology.
Yeah, those are great examples. Yeah, there's many ways that mRNA is going to help. Maybe in I think the list is enormous, but just to go through some of these. Yeah, so we talked a lot about using the liver as a bioreactor for protein replacement and for fixing proteins, as well as in cell therapies, so modifying cells ex vivo, and that has some big advantages over viruses. You'll be able to keep those the viral components out, and instead using lipids and mRNA instead, and gene editing, and as Dr. Dong and Dr. Hedrich mentioned, is a lot of that, in the future, I think will rely on mRNA being able to deliver the gene editor, the base editor, and yeah, and of course in vaccines, we'll continue to use mRNA for infectious diseases, as well as for cancer vaccines. And of course, the advantage there over DNA-based methods is just the rapid speed of development.
Yeah, I'll just add, I think for the cancer vaccine part, there are very exciting clinical data from Moderna, from BioNTech. So probably there would be some very important cancer vaccine would be developed in the next few years, integration with current immunotherapy with PD-1 or some other immunotherapy altogether that make cancer maybe curable.
All right, probably I will ask a question from the audience. So I think we talk about the liver. I think most of the mRNA therapeutics, they're mainly targeting the liver. So liver is the major organ. So one of the question is that has decreased or changed delivery efficiency or efficacy in mRNA lipid nanoparticles being observed in the liver dysfunction population or animal model, and will that cause an issue?
That's really a great question. We at Mount Sinai has a very strong liver program which study liver cancer, liver fibrosis, and many other liver-related indications. And after I joined here, I really learned that in the liver, there are so many different cell types. So it's not just the hepatocytes, not just the endothelial cells, epithelial cells. There are many different cell types, even in the normal physiology, and also there are a lot of different diseased cell populations, such as liver cancer cells or fibroblasts for fibrosis. Definitely, we notice the delivery would be very different for normal cells versus disease cells. But it's case by case, and also for different lipid nanoparticles or any type of particles, they would perform very differently for all these different cell types.
Great. So we'll ask another audience question now. So there's actually two that are quite related, and so maybe we can group them together. So what are the various strategies that can be opted to mitigate challenges associated with LNP delivery to the lungs, especially in regards to mucus penetration? And the second, I think, very related question is, do you have an optimized LNP formulation to treat CF diseases?
Yeah, these are very good questions. So the short answer is yes. So you really have to optimize the lipid nanoparticle formulation. And what we've learned, we have done a lot of head-to-head comparison between different epithelia, skin and lung and other cells, epithelia as well. And it's really striking that it varies a lot, you know, the editing efficacy. So one lipid nanoparticle surely doesn't fit all. So we have dedicated quite a bit of time on trying to identify the ideal ionizable lipid. So at least in our data, or based on our data, I can say this seems to make the most difference, honestly, and that, you know, changing and optimizing the ionizable lipid and the pKa can really bump up editing efficacies a lot. So this is one. On the other hand, when it comes to mucus penetration, that's the other angle, right? So on the one hand side, you need transfection of the cells, but the mucus is a completely different story. And there we have teamed up with companies like Acuitas, and together with them we have identified some modifications that we introduced to the PEG shell of lipid nanoparticles that make them moving very efficiently and fast, even in cystic fibrosis mucus. And so this is quite promising. So we are trying to take that further and combining that with the optimized lipid nanoparticle formulation, hoping that by combining that, we can, on the one hand side, traverse the mucus efficiently and also transfect and edit the cells.
Thank you, Sarah. So physiological barriers such as mucus is, of course, one of the main challenges for mRNA delivery. So what are the other major challenges in the field today? Can you comment on this?
One for sure is the half-life of expression from the mRNA. I mean, in other technologies such as gene therapy based on adeno-associated viruses, AAVs, I mean, those can theoretically last for forever, or at least years or decades. But yeah, mRNA in many cases will only last for about a day or so. And of course, all the advances that have been made in mRNA to stabilize them, modifying the uridines on the mRNA, all of these that led to the Nobel Prize for mRNA. I mean, these are all positive things. So being able to continue to increase the stability of the mRNA, prevent from being degraded and express proteins is one of the major barriers. And things like making circular RNA, making other modifications to nucleotides, self-amplifying RNA. These are all viable approaches to extending them.
I'll just add, I think for the delivery system, we really need to develop a more biocompatible and safe delivery system. I think based on current recent clinical data, there's a certain level of side effects from delivery system, and some companies, they need to change it to other or newer generation of the lipid nanoparticle to reduce potential side effects and then increase the therapeutic window or therapeutic index. That's really important for, I think, any type of indications.
Great. So I'll go ahead and ask another question from the audience. So they say, thanks for the great presentations. My question is regarding the cardiovascular diseases. So I wonder how much modifying the structure of different lipids in the LNP, adjusting their molar ratios, or surface modification could be effective to treat cardiac tissue specifically.
I'll provide a little bit. I'm not sure I have the perfect answer, but there definitely is some transfection of the heart with lipid nanoparticles when they're injected IV. It's not a lot, but there is some transfection there. But what's not really known, at least I haven't seen the data, is what happens for like in diseased tissue. So if there is there's ischemia, there was part of the heart that was damaged, the data might be there. Again, I just haven't seen it. It's not really known to what extent lipid nanoparticles mRNA can be delivered to those. And I think those are really important questions. Not just how it's delivered in for healthy individuals. So when there's areas of vascular damage, when there's tissue, there's inflammation, how it happens there. So at the moment, I think in order to improve cardiovascular disease, the way to do it is to modify the liver to change the blood, and then have the impact on the blood, which then helps in various cardiovascular diseases. But like was asked by the participant, being able to directly go right into the heart, the blood vessels, other is really the future, hopefully the near future of the field.
Yeah, and maybe I can add to that while it's known that modifying lipid nanoparticles, adding certain lipids or removing others or modifying the ratios, there's a lot of data that by doing this, you get a certain tropism of the lipid nanoparticles. But overall, I always have the feeling that it's still not fully understood, right? So we know, okay, when adding charge, we can get it to the lungs more than to the liver, for example. But that's basically all we know, right? So actual targeting strategies, understanding them in full detail, I think we are not there yet, and there is still a lot of room for research and for improved understanding.
Great. So a couple of days ago, Moderna's another vaccine, RSV vaccine, received FDA approval. Now we have a couple of mRNA vaccines have been approved for human use. So what application do you think the first mRNA therapeutic will be approved for?
I think probably cancer vaccine, right? There's already very promising clinical trial data from phase 1, I think now phase 2. I think a cancer vaccine might be the first one.
Yeah, I'm not not sure where it lies in the pipeline, but one that I think we're all really excited about is gene editing to modify proteins in the liver, like Verve's PSK9 gene cutting strategy. So I think it's Dr. Dong said, it's these are further away from being fully approved, but ones that are in the pipeline and presumably will be approved if a couple issues are sorted out.
What do you think, Sarah?
I'm with Yizhou on that. I hope it's the cancer vaccine. I mean, I think this week Moderna released or presented the data at the ASCO conference, right, in the US, and it's pretty impressive data, and it's a very much needed therapy. So I really hope that that they will be able to get approval for that very soon.
Mm-hmm. Great. I'll ask another question from the audience. So they say, thanks for the interesting talks. I recently attended a microfluidics workshop where I learned how useful multi-organ on a chip devices can be for testing the organ toxicity of small molecule drugs. Have any of you used these techniques for screening these large lipid encapsulated RNA-based drugs?
Maybe I can answer that, because my lab is developing such models in order to test new treatment options. And I'm actually not aware of any, at least published data, where nanocarriers or lipid nanoparticles and mRNA therapies have been tested in a multi-organ chip setup. But they are definitely suitable to do that, and that's the vision of the future as well. That basically you can emulate topical application, but you can also inject it systemically into the microfluidics and then monitor where the lipid nanoparticles accumulate, if there are certain side effects. So that's really the idea for developing those organ-on-chip setups. And they are definitely not only suited for small molecules, but also for next generation therapies.
Great. So I will ask another general question about the stability of mRNA, because I remember like at the beginning of the pandemic, the storage and the transportation of the vaccines require like a -80 freezer. So what advances in the field have happened since then to improve the stability of mRNA?
I think definitely there are a lot of improvements. I think even during pandemic, Moderna or BioNTech, they have performed a lot of stability studies, and later on they can store the vaccine at -20 degree. And more recently, I think there are quite a lot of published papers. Some that maybe I don't know how, not much details on that, but they presented they can store the mRNA vaccine at a room temperature for quite a few weeks. Definitely, there are more and more studies on stability, either using different formulation components or using some excipients to stabilize both the particles and the mRNA.
Great. So we have another question from the audience. So we've talked a lot about some different tissues, but they're wondering if there's an optimized LNP formulation for delivery to the brain.
Yes, definitely brain is most difficult organ to deliver because of blood-brain barrier. We have been working with several biotech companies, try to work on lipid nanoparticles to cross BBB, and we hope we can publish data and release that in the next couple months, and there will be some very exciting results on that.
Yeah, and my impression is that that's where the state of the field is at the moment. There's some there's a little bit in the literature on being able to do an IV injection and transfect the brain, but it's very limited. I think, like Dr. Dong said, is perhaps the most limited data on various organs. But there are things being done in laboratories around the world. So there's definitely a potential there and promise, but it's at an earlier stage to get some mRNA into the brain and be able to express proteins. Intrathecal injections can be done. So IV injections are challenging. Intrathecal, being able to deliver right to the CNS is another way to do it.
All right. I will ask another like a general question about the mRNA therapeutics, because Dr. Dong mentioned like right now one of the focus on lipid nanoparticle is further improve the biodegradability and improve its biocompatibility. So what can or has been done to reduce the side effects of mRNA therapeutics, including from the mRNA and also lipid nanoparticle perspectives?
For mRNA perspective, I think the Nobel Prize is a great support for that. There's a chemical modification of the messenger RNA that would reduce non-specific immune responses or immune stimulation. For the delivery system, there are several components: ionizable lipid, helper lipid, cholesterol pack. All those are very important. I think, as Sarah mentioned, ionizable lipid is a key component that may trigger certain type of side effects. And also the overall formulation is very important. The ratios for the components, and even like we think phospholipid is very biocompatible, very safe. But if you use different ratios with same particle, that may also cause some different side effects. I think all those lipid chemistry, formulation components, ratios are all very important. And also even PAG, it's small percentage of PAG, but researchers found there's an antibody against PAG detected in mouse models or even in, I think, a human serum. Those need to be carefully studied in future projects.
Maybe add to that with regard to the mRNA. I mean, currently, really, the mRNA design is very universal and not tissue specific. So most of the mRNA designs are tailored towards vaccination, for example. But what we looked at how the different tissues and epithelia, in our case, respond to mRNA, and they have completely different responses. So whereas the skin really doesn't do much, it doesn't seem to care, the lung really is fully activated and they ramp up all the interferons, what they got. So I think, there is a lot of room for improvement and to generate tissue-specific mRNA, which would eventually then also help to cut down the side effects.
And I just want to emphasize that there's definitely room for improvement. I think we're starting from a great place. Just for example, with Onpattro, the siRNA lipid nanoparticle drug, it's using a relatively similar lipid nanoparticle format to what's used in the mRNA, and of course it's also RNA. And patients have been injected every 3 to 4 weeks with this therapy. I think going on 10 years now without major problems, as far as I know about. So I think we're starting from a great place. Delivering RNA with lipid nanoparticles has a great safety profile so far.
Yeah, thanks for sharing those perspectives. So I know we're running short on time, so maybe we'll just do one last question. This is a question from an audience member who's wondering, antibody-modified LNPs are popular in preclinical studies. Do you think they're promising for translation in terms of CMC? So do you think they're scalable, translatable to the clinic?
So maybe an unpopular opinion, but my guess is no. It's probably too complicated. I mean, it's, when you then think about translating that, industry is looking for simple solutions, and attaching an antibody to lipid nanoparticles is quite messy. It's quite challenging. And then also the quality control to make sure how much is there, where is it located, will it stay there? Scientifically, I think this is super exciting and worthwhile exploring, but I have my doubts that this will be like, a major thing in the future. But maybe I'm wrong. Let's see.
I'm a little bit more optimistic. I definitely think it's very challenging for the CMC of this type of complex system. Meanwhile, I feel if there is a urgent need for certain therapeutic indication, currently there is no any other treatments, I think engineers may figure out some way to make those happen.
Christian, do you want to add some comments on this?
No, thanks.
I want to extend my heartfelt thanks to our panelists for sharing their expertise and insights. It's clear that mRNA therapeutics holds a tremendous promise for the future of medicine. And as the research progresses, we can anticipate even more innovative applications and advancements in the field, leading to new treatments and improved health outcomes for patients worldwide. I would like to also thank the Gairdner Foundation for organizing this important event and for their continued support of scientific research and innovation. The Gairdner Awards have recognized and celebrated groundbreaking discoveries in biomedical science, and this panel is a testament to the foundation's commitment to fostering scientific dialogue and advancing knowledge. So thank you everyone for joining us today. Hope you enjoyed today.